Bueno, muy buenas tardes a todos. Mi nombre es Susana Koppelman.
Good afternoon, everyone. I'm Susana Koppelman. I welcome you on behalf of Cegyr to our third virtual tribune in our fourth exchange session and the last. So a fourth session. It's going to be very interesting, and today we are going to share experiences and ideas with two outstanding professionals from the Huntington Group, Brazil, and we really thank you for sharing your time and your expertise. Our doctors will give two presentations, followed by a panel of experts, and they are going to make your contributions. And we thank the audience for sending your questions through the Q&A function. Dr. Kosia and myself will be broadcasting your questions. Another reminder, our doctors will give the presentations in English. You can listen to them in English or Spanish. You can choose your audio language by clicking on the interpretation icon, so you can suit yourselves. So our final session for this year, the last but not least important. Thank you, doctors. Dr. Andrea Kosia, my partner in crime in coordination. Dr. Kosia is a specialist in reproductive medicine. She's the chairwoman of the fellowship program of assisted reproduction, and she's the coordinator of the cryopreservation program. Andrea, hand over to you so you can introduce our panel of experts. Thank you, Susana. Thanks, everybody, for joining us. A pleasure to share this moment with you. Thanks to the audience that were loyal to the end. And I have the pleasure to introduce our speakers. First, Dr. Thais Sanchez Dominguez Uri, senior physician in the Huntington Center of Reproductive Medicine. She's a specialist in assisted reproduction with a PhD on polycystic syndrome and assisted reproduction. She is the coordinator of the ovocyte donation of the same institution. She's a board member and she's a partner of this institution. She's going to talk about embryo implantation, how to improve results. It's a pleasure, doctor. Hand over to you. You have the floor. Thank you, doctor. Pleasure.
It is a pleasure, and I want to thank all of the Cegyr group and the scientific committee. So I'll share my screen. Can you see my presentation?
Yes, perfect.
Yes, okay. So let's start now. I'm supposed to talk about embryo implantation. How can we improve the results? And for sure, this is the answer that all of us and our patients want to know, at least over the last decade. So now, today, I'm trying to manage these expectations and maybe bring some hope for now and the future. I have nothing to disclose. And speaking of managing the expectations, our reality shows that early reproductive failure is the most common complication on pregnancy. Human reproduction is surprisingly inefficient when compared to other mammalian species. More than 70% of conceptions cease to develop, and the vast majority even achieve clinical diagnosis. So the implantation failure is assumed to be related to the impairment of the embryo development potential and or the endometrial selectivity or receptivity. And since Louise Brown's birth, the gains on assisted reproductive technology has allowed many couples that previously are unable to conceive to achieve a viable pregnancy. Since the ICSI that helped lots of men with severe male infertility, passing through the oocyte cryopreservation, helping our oncofertility patients, and now with the last achievements on technology with embryoscope, time-lapse, artificial intelligence that Catherine will talk later, we have improved at least the chances of getting pregnant. But what about the rates? We still keep the same chances that we achieved a decade ago of having a baby at home, around 30 to 40% of live birth rates. And during this fertility evolution, although improving overall outcomes for these struggling couples, a new challenge has emerged: implantation failure. Both patients trying to conceive, either natural or artificially, can suffer from implantation failure. But recurrent implantation failure is related only for those undergoing ERT treatments. Although there is no accepted formal concept on the diagnosis of implantation failure, most authors define as more than 3 attempts of transfers without any success. Trying to be more precise, some authors adds maternal age, number of good embryos, but due to all these variations in definitions, there are scarce data that accurately represents the real incidence of prevalence of this fact. A recent publication showed that we can expect the same chances of pregnancy per transfer of euploid embryos after 3 attempts, that is around 60%. And deal after 3 transfers with less than 5% of RIF. I will speak RIF because it's easier, recurrent implantation failure, so I'll tell RIF. But we must have in mind that this paper selects only good prognosis patients with less than 35 years and with at least 3 good quality embryos. So this reality is not for everyone. And considering the fact that after hatching, the formed embryo has to attach and invade the endometrial layer, we can imagine that the most common factors responsible for these failures are supposed to be related to the embryo itself, to the uterus, or even with the maternal environment. And this is a little bit about what I'm telling you today. Some of these can be considered as risk factors, and we can at least try to control them and to advise how to improve our results. And I like very much this sentence because I know, at least if not all of you, the majority agrees with me. It's much better to prevent than to treat. So some things we can prevent. And I'll start with the maternal factors. And a lot of them will be related to the embryo quality. And of course, I have to start with maternal age. Maternal age is the more important fact and risk for sure of among all of women. We know that our decision to postpone the onset of motherhood, the onset of maternity over the last decades lead us with the increasing number of infertility. And nature didn't understand this decision of a lot of women and continues to make us lose not only quantity, but also quality of our eggs. And quality maybe is a more deleterious in the end that women face, as shown by the group of Monet some years ago. Despite increasing the number of embryos, the aneuploidy rate is directly related to women's age. And with this fact, we cannot do anything about it unless advise our patients. And this data is showing year after year to all of the societies and all of the authors. And this leads the women to face the great risk of infertility during the years. So it's our duty as specialists to counsel and provide information. Since the gynecologist is counseling in their offices, and us specialists and the societies, we have to do some campaigns about it. We have here in Huntington, this one that is called Trintou Congelou. This highlights to all women that around 30 years, they have to check on their fertility. And if they want to postpone maternity, they have to do something. And what we have nowadays, we have the oocyte cryopreservation. But despite this fact that we cannot change a lot of things, there are some things around women environment that we can change and we can modify prognosis, such as those around lifestyle. Increased BMI, not only for those women who has to do the ovarian induction, they need more gonadotropin and they will face less oocytes. So it's hard to select the better embryos because they have low amounts of embryos. And this is probably related to the oocytes, not with the uterus, because this recent review shows that among women, among recipients from donor oocytes, they don't face less rates of live birth. So probably this is related to the embryo quality. Another thing is smoking. Despite it's well known that smoking has better prognosis, a worse prognosis with pregnancy, leading to high miscarriage rates, less oocytes, and increase on FSH is what women that smoke face too. But still the relationship with RIF is controversial. But this is an easy thing, maybe not so easy for the women to do, but it's an easy thing for us specialists to counsel our patients. Talking about clinical factors, it's well established that thrombophilias are linked to worse obstetric prognosis and complications, mostly recurrent pregnancy loss. Usually, acquired thrombophilias are related to this worse prognosis, but with recurrent implantation failure, we don't have the same relationship. And as all of the clinicians, the gynecologists counsel their patients, we consider that the TSH below 2.5, it's good to have a good ovulation, a good oocyte quality. It not depends on less than 1.5 to at least less than 2.5. It's good for the patients to have a good ovulation. Okay, now let's talk a little bit about the uterus, the uterine factor. Starting with the gynecological factors. And here again, we face other controversies on literature, mostly due to the sample size, small sample size that analyze RIF on these patients, and the bias analysis also. But this is a good paper with a large number of patients analyzed, and despite being a retrospective study, they showed that only the submucous fibroids are related to recurrent implantation failure on the right side of the table, nor polyps or adhesions. So we have to be careful when we analyze and when we diagnose these things on patients, and we have to be careful to advise which patient needs to be searched to have this direct exams to do this diagnosis. But most of societies, despite a lack of evidence, advise and consider that we should be reasonable to do septoplasty and polypectomy, considering the women prognosis and what have she passed through and suffered in other pregnancies or attempts. We can check also for chronic endometritis, and we can see through hysteroscopy, we can look for the structural aspects of the endometrium, the presence of micropolyps, but all of this, as we all know, has a low accuracy on the diagnosis of chronic endometritis. And the gold standard come through immunohistochemistry and the diagnosis of CD138. Some paper shows also that we have positive bacterial cultures on around 70% of patients with recurrent implantation failure, but this is not related to chronic endometritis. So, in this way, on the omics field, authors are searching for the ideal method that describes the endometrial microbiota on patients with implantation failure, not only with RIF, but implantation failure at all. Looking for the presence of pathogenous RNA or absence, lack of lactobacillus. And despite the increasing number of studies, we don't have a consensus of which the sign is reasonable to consider and what is the ideal treatment. Some other gynecological factors as adenomyosis and endometriosis can worsen prognosis on embryo implantation. Both can be accompanied of an increased level of estradiol, a decrease on progesterone levels, and both these two signs are associated with a modulation of the immune system. And this disruptive immune system can explain the high rates of inflammatory and low implantation rates. So, how can we try to improve the results? Of course, thinking of the endometrium, it has to be a right and good balance between selectivity and receptivity, leading to not perfect, but at least near an ideal window of implantation. And this insane search of the ideal window of implantation for more than 80 years since Rock described anesthesiological changes during the implantation time in the endometrium leads us today to the latest omics research as the microbiota that I told in the other part. The endometrial receptivity array test initially evaluated the endometrial biopsies array expression in array expression for over 200 genes. Now we have more than 3 commercial platform arrays that promises to evaluate the individual and the exact time of transfer, and thus synchronize the embryo with the precise and individual window of implantation for that woman. But is this the way, the path? We can achieve better results with this exam. Despite the clinical large trial showing these benefits, or at least suggesting these benefits, other studies fail to achieve the same results for all infertile women. So maybe this is a way, this is a good thing for us to search on patients with rift, not for all infertile patients on the first failure. And the immune factors. As the same trying the same attempt to achieve the perfect window of implantation, now there is more data around the uterine natural killer cells. In terms of allorecognition, interactions between members of the killer immunoglobulin-like receptor, the KIR, expressed by the uterine natural killer cells that will bind to the trophoblast human leukocyte antigen C, the HLA-C, can be deleterious because both are highly polymorphic. So if we have a woman with KIR AA and the fetus, the embryo actually, with the HLA-C2, maybe will not achieve a good implantation rate because this combination diminishes some growth factors, some cytosines related to a good implantation and to a lower inflammatory endometrium. And then this disbalance we can find in other uterine disorders, as the adenomyosis, endometriosis, and chronic endometritis, as I said before. All of this dysfunction leads to less production of epithelial growth factor, metalloproteinases, cytokines, and growth factors that are essential to a good implantation. This is a great paper because till now we don't have which is the better test to perform in our patients and to check for these immune factors, and even less, which is the best treatment to provide for them. So Nathalie Lédé have published another paper in 2020 that shows that not only the presence or absence of the uterine natural killer is important, but the relationship with the cytokines and the immune cells production. So this expression leads her to separate the RIF patients in groups. And she suggested some protocols using all of these therapies, endometrium scratching, high doses of estrogen, corticoids, HCG, sexual intercourse, immunotherapy, either with immunoglobulin or intralipid injection, and combine all of these treatments in a way to improve the low immunity activation, so to try to provide higher levels of all of these proteins and cytosines, or diminish the inflammatory milieu on the patients that have overimmune activated endometrium. And if we have endometritis, this recent meta-analysis shows that treatment with doxycycline for 14 days as first-line therapy, improves not only the implantation rate, but the clinical rate and the live birth rate also. And now I'll come back again to the age. This is quite boring for our women when we start talking to them about age, but it's clear that today our counsel is different from our mothers. At that time, there are counsel about contraception. And now we need to counsel our patients about conception. Not only if they want to have babies or not, but if they want only to think on the future after 30 or 35 years old, if they want or not to become mothers. And if they want to have 1 kid, 2 children or more. This is quite important to do nowadays. But if we don't have this opportunity, and as the majority of cases we face are women above 37 years, we have to come back again to this data published by Monet and see that despite worse outcomes identified in older women, the invasive way to select the embryo according the ploidy of the embryo, we can selective transfer a euploid embryo and improves at least that embryo transfer performance and improve the age-related decline of the embryo implantation. And as we all know, maternal age plays a crucial role in the quality of the embryo. Not only the ploidy, but the quality. There are a lot of studies that compare the implantation of a euploid embryo for women below 37 years and those above 38 years. And we can see worse results for those above 38 years, even with euploid embryos. So sometimes we'll have to face the reality and make our patients to understand that we need to move on for another option and choose the oocyte donation instead. Keep chasing miracles. We have, as you have also, a good program of oocyte and embryo donation. And sometimes we have to move on to this treatment. And it's a great treatment. It's not a lack of opportunity. It's a great opportunity. But not all women, the embryo is not so good because of age, because of aneuploidy. And we have good tools to do the embryo selection through a non-invasive way. It's the time-lapse incubators. I think maybe I have a video here, but for sure, Catherine will talk about this so much better than me. And we can see through the embryo development during some specific times if we have a better or worse embryo, and we can for sure help our embryologists to select the best embryo for the transfer, and this trying to improve the outcomes. So we can add these images, we can add this information With other parameters, we can associate morphokinetics evaluation with patient cycle parameters. Maybe on the future, the artificial intelligence will tell much more about the embryo quality and the embryo selection for us. Our group is deeply studying this issue that we were orally presented on the last session in ASRM. Catherine is on the right side. She presented on ASRM. And this paper showed that we can improve the embryologist's accuracy on selecting the best embryo and manipulating less also the embryo. Okay, but till now, we don't have any miracle. And I think we all need to move forward and to look for the future, hoping and expecting some better plans and better tools and better ways to individualize and treat our patients. Till now, we know just a little bit or nothing about the embryo and uterus crosstalk. And maybe this is the future. Some papers published on Nature this year, they showed us, this year and the last year also, they showed us that we can use organoids in an attempt to understand this crosstalk. These organoids are structures that came from pluripotent cells, and we can achieve the epithelial and stromal fraction of the endometrium. This is called the assembloids. And with this, we can study, we have a model to study the decidualization and the implantation. And we can also, with these organoids, understand a little bit better the embryos, and we can provide the blastoids. And this interaction maybe can tell us in the future what is the correct tool and the correct way and manner to deal with each and every patient. So I want to leave you with these take-home messages. Despite having a great team of embryologists, nurses, physicians, we have always to provide the psychological support for all of our patients, mostly for these RIF patients, because around 5% of them did not realize their family goal, mostly due to the treatment dropout. So we have to care about them. We have to do individual diagnosis and treatment, and we have always, whenever possible, prevent at least these things that we can prevent and we can early diagnose and manage. We have some steps going to the artificial intelligence that Catherine will talk much more for us. And a possible future with this is to know and to understand better the in vitro models, the individualization of patient care through these in vitro models. Thank you very much for your attention, and I'll be pleasure to answer all the questions in the end of the presentations.
Bueno, muchas gracias, doctora. Excelente presentación, muy interesante.
Thank you so much, doctor. Brilliant presentation, very interesting.
No tenemos dudas de que luego va a haber preguntas.
There is no doubt that there will be plenty of questions later on, so we urge our participants to write their questions in the chat or Q&A box. We will continue now with our next presentation. It is my pleasure to introduce Dr. Catherine Jacobs. She's a senior embryologist at the Huntington Center for Reproductive Medicine in the gene group with experience on embryonic assessment with time-lapse and blastocyst biopsies. She's part of the scientific committee at the center, and she's working with artificial intelligence to improve embryo selection. Doctor will be addressing us on artificial intelligence and time-lapse. It is a pleasure to have you with us. Thank you so much. You have the floor.
Yeah, it's my pleasure. Thank you for the invitation for this exchange session. Let me just share the screen. Is it fine?
Yes, yes, yes.
Yes, okay. So I find this idea of the exchange session really nice. I know already Florencia, and I know that you also have a time-lapse system. So it will be very interesting to tell you a bit about what we changed since we started using these time-lapse incubators, and also how we came to use and maybe fully apply the artificial intelligence in our routine. I'm an embryologist, so I will be talking more about the embryo selection and this part of the use of the artificial intelligence, but I think it will be really nice. And so this whole lecture will be about the new road that the use of these incubators, like the EmbryoScope, brought us. So switching from these benchtop incubators, where we had almost no information about the embryos, to this one that provides us endless informations, so things that we are still actually learning about it. So what did really change? We can start talking about what everybody thinks when we talk about time-lapse. So instead of having static images, 3 or 4 images from the whole development of an embryo, now we have a whole video. So it works as a picture movie. So we have pictures taken from the embryos every 10 minutes and from different focuses, and taking them all together in the end. So 5 days of development can be seen in seconds, like you can see in this video from the right. That is from an embryo since fertilization until day 5, when they reach the blastocyst stage. And besides thinking always about this evaluation part, I think that when these incubators, they were put into practice, what was always thought was, can we benefit really from it? And this is also much discussed nowadays. And we see in every Congress, all these big conferences, the ASHRI and the ESRM, there is always a session debating if there is in fact a benefit in using the time-lapse in our routine. And actually what the systematic reviews and the method analysis show us is that it hasn't been proven yet that it is really beneficial for the patients. So there is low to very low certainty in the efficiency, in the effects or the benefits of using time-lapse. I personally disagree with it, and I agree with what was said in this letter that was sent in response to this specifically systematic review, that actually we need further well-designed studies to really be able to establish and delineate the clinical utility of the time-lapse system. And I would like you to take your own conclusions, and then in the end of the presentation, we can talk about it. But talking about it just as an incubator, the idea is that we remove the dishes with the embryos less than we were used to do. So just taking that in mind as a first step, when we culture the embryos in the time-lapse system, we reduce to 0 the exposition of the dish during the whole the embryo development. And from the conventional incubators, we already had studies showing that just keeping them there would improve the blastulation rate and the embryo quality. So from that point, we already had a plus or a point to show that actually this incubator can be beneficial for the embryos. And that was the first study that we did in our clinic once we started using these incubators. And we actually saw an improve in the blastocyst formation rate after the 2 PNs. So from every 2 PN or for every 10 2 PNs that we had almost 10% more blastulation. And for the patients, when we divide them in the age groups, from patients between 34 and 42 years, we saw that they had in the end of the cycle more or less 1 extra blastocyst. So this was probably due to a lower arrest rate that we saw in this incubator. And I cannot assure you that this is because of the uninterrupted culture, or maybe it is because of the evaluation that is completely different. So this is also something that we have to keep in mind. It's very hard to design these studies comparing it because you really evaluate them completely differently. Maybe you also have it in your clinic that you might think more about leaving the embryos that are in the embryoscope until day 7 than the ones that are in the bench tops, because you actually see that they are growing a little bit. So let's wait a bit more. And sometimes they are just slowly, but they get to the point of becoming a blastocyst. And besides that, we can also think about how inconsistent our evaluation in the conventional way is. Some studies have shown that we are generally good, but not optimal in this agreement. Not only between embryologists, but also the same embryologist would evaluate an embryo differently in different days. And imagine if they only had one check, a quick check in the microscope. So this is really very inconsistent. And we usually only had the, I would say, 3 days of evaluation. That's what is the most common routine for those that culture embryos until blastocyst. That will be the fertilization, the number of cells in fragmentation on day 3, and then the Gardner evaluation for blastocyst. So blastocyst size, true ectoderm, and inner cell mass rate. And when we talk about the time lapse and the morphokinetics, we don't have only these 3 days of evaluation and these few parameters. So now we talk about all the cleavage timings, all the multinucleation that we can see in 2 cells or 4 cells. If this embryo has some abnormal movement, something like direct cleavage or reverse cleavage. And from this point, you already have much more data to analyze. And you can also move the video forward and backward and call your friend and the other embryologist to see what they think about it. So we have seen that with the morphokinetics annotations, embryologists are much more, they agree much more between themselves, and the level of concordance comes to high perfect. So actually, let's talk about what do we annotate there. So this is like what we see in the embryoscope. I think that you also have an embryoscope, so it will be very similar. We actually we annotate how many hours or minutes after the fertilization an event happens. And from these hours that are annotated, there is an algorithm inside the computer that gives you in the end of the development a note. So you have an idea of how good that embryo is, besides only knowing the cell quality. And so we have these timings, and we also have these abnormal cleavage that I told you. I will say tell a little bit more in the next slides. We also see not only the times, but you can also calculate how long was a cell cycle duration. So there are many possibilities. And besides what comes with the software, you can also build your own software. and see if you have a different result or what matters for you in your routine. The abnormal events that I told you would be some of them. I have some examples here. So in the first picture, you see a little third PN that appears and disappears that you would have missed in a conventional evaluation. The second one, we see a failed division. So the oocyte tries to divide, or actually it moves like it was going to, and then it comes back to a one cell stage with multiple pronucleus, and then it goes beyond to 2 cells. Then we have also the direct cleavage that was something that made us very crazy because one cell goes to 3 and or to 6, like you can see in the other one. And if you just see a static evaluation, you would say that that embryo is pretty fine. And this amount of data, they have been studied they have been studied by researchers in the whole world since the beginning. So trying to find predictors for blastocyst development, for blastocyst quality, if they were correlated to implantation and ploidy, and even to the live birth ending. But to build that amount of data to get to this point of research, it takes a while. And that's why we always begin with what is the probability of having a blastocyst since we have these cleavage timings. And then we move to ploidy, and then we move to implantation, and then finally we get to the clinical pregnancy and the live birth. So it's been a path that the whole world was going actually more or less in the same time. They were all implementing this technology and trying to figure out what it meant. So here you can see it. It's a very good example of it. And this new concept that we have, all these morphokinetics data, they brought us some disagreements. So in each line, we see one study, and then we see when they had some significance, that is the green lines, and then some groups find the significance in the PN appearance and PN breakdown, for example. And some others don't see any difference on that in the embryos that implanted or didn't implant. And it happened with several groups, and then they all together had to stop and think, well, so maybe we are not finding one parameter that will show us if an embryo implants or not. And maybe we cannot copy or use what another group find out, and we should see in our reality what applies. And besides these disagreements, there were also some agreements that were found out. So here I brought you the Istanbul consensus that had the fertilization check, the ideal time to be analyzed at 17 hours plus or less 1 hour. And actually we saw that analyzing almost 8,000 embryos, that the ideal time to do this check would not be 17, but 16.5 hours and half an hour more or less. If you go further than that, you would be missing many pronucleus. And another very well established concept was about the 8-cell stage embryos, that if an embryo had more than 9 cells in the 3rd day, they would be considered accelerated. And these fast embryos would be judged as not as good as the other ones, because they would have higher incidence of chromosomal abnormalities. And this is not what we see nowadays. So in a summary, I would like to tell you that, well, we have tons of things that are done differently in the laboratories. And besides all the studies that we have, sometimes what applies to one might not apply to the other one. And what and how to annotate these parameters. So here we started with the parameters that the EmbryOscope asked us to annotate to give us a score. And then later on, we decided to mark almost all the possible parameters that the EmbryOscope had, so that we could see what for us was indeed important. And I will already tell you that in advance that we nowadays, after all the studies that we have done, the parameters that we annotate as are these ones that you can see on the right. So the number of PN, time of pronucleus fading, then the time of reaching 2, 3, 4, 5, 8 cells, time of bustulation, so when the embryo reaches all the zona pellucida, and then the inner cell mass and true for ectoderm quality. These are the parameters that today, nowadays, in this moment, we have defined that are the important ones for us. But, and I know that it takes time and it's very demanding, but maybe it would be the ideal world for all the clinics that have these systems to do. So, as I told you, we started with all these parameters, annotations, and this, of course, brought us to tons of data. So this that you are seeing is actually the Excel sheet that we can export from the EmbryOscope, and all this data, it means a lot. So we have to stop and see all that it brought to us, and our studies were then trying to understand them. So the first one was actually just to correlate these parameters with the blastocyst quality. Then a little bit later on, we found out what were the parameters that could predict the aneuploidy. And as I told you, that concept that the 9 cells embryos were accelerated and tended to be aneuploid, for us was not the reality. And actually for all the groups in the world, we found out that that was a misconception. So in fact, when we talk about morphokinetics and genetics, the faster the embryo, the better it is. And for us, the parameters that we found to be relevant were the time of pronucleus fading and the time to reach blastocyst in that first analysis that we did. And it's interesting, and I would like to spend a little bit more time talking to you about the morphokinetics and the genetics, because in fact, how do they correlate? If we are talking about the timing of the cleavages, we are talking actually about the timing that every cell cycle takes. And for one cell cycle to be completed, we have to have the correct DNA synthesis and the segregation in the cells. So the faster the better, but why? Let's talk a bit about the cell cycle. And I would like to tell you about the spindle checkpoint. It's very interesting to think about it so that we can understand why one embryo is slower and why one embryo will be faster. So in the cell cycle, it is in the metaphase that the chromosomes are condensed and aligned in the mitotic spindle. And in this moment, we have this spindle assembly checkpoint. So if every chromosome is correctly aligned and attached to the microtubes and centrioles, the sac allows the progression to the anaphase and telophase. But if something is wrong, this is the system that will arrest the cell cycle progression until all the chromosomes are correctly aligned and attached to the microtubules. There is this really nice study that was published in 2017 that shows us what happens until day 3 in an embryo. So this embryo, so until day 3, they have this sac. So the system is there, but it's not totally functional. And this could be one explanation for the abnormal mitosis that we have that lead the embryos to aneuploidies. Or to those cells that we see that they are so slow, they stop, and then we see another nucleus inside. So in this case, the interface restarts without a cytokinesis. So new DNA is synthesized, but the cell doesn't divide. And that's one scenario. And the other one is that the embryo can continue to divide without correcting what was wrong. And so it divides fast, but with aneuploid cells. And that was the scenario that was shown in all these studies that said that embryos with 9 cells were fast. So there were studies with blastomers and fish analysis, so only day 3 embryos. And when we move to after day 3, we see that actually after day 3, the system is present and functional. And then what we see is that the cell cycle progression is arrested until the repair. Or if this repairs doesn't happen, then the cell might go to apoptosis. That's the reason why we have less blastocysts, and the percentage of blastocyst ploidy is higher than the day 3 embryos. And this could also explain why those embryos with direct cleavage in our reality and also in some other studies, they do arrest much more than embryos without this direct cleavage. That's what we see in the left. So the embryos without direct cleavages, they go to blastocysts in 64% of the cases. And the ones with direct cleavages, they are lower. So 14 and 43% of them reach the blastocyst stage, depending on the moment that this direct cleavage happens. But once the embryo reaches the blastocyst stage, their ploidy rate is the same as the one that didn't have this cleavage, this direct cleavage. So that's very interesting and could be an explanation to what happens. And that's seen in all the studies that show us that after day 3, the correlation to aneuploidy is seen actually in the later parameters. So start of blastulation and expanded blastocyst and even blastulation timing. So let's go a little bit deeper on morphokinetics and diploidy. I would just mention to you what this review showed us. It studied more than 40 embryos, and it correlated all the parameters in subgroups to see which ones we could relate to aneuploidy or not, okay? So, of course, I would just like to say that there is an overall moderate risk of bias. So what we see here might change in the last years. This it's not consolidated idea yet. But for multinucleation in the 2-cell stage, there was no association to ploidy or aneuploidy. But if the embryos had multinucleation in the 4-cell, they found out that it's a higher chance of aneuploidy in 13 studies. 10 studies showed them that reverse cleavage, when it happens, it's actually more propense that that embryo is euploid. And in case of direct cleavage, those blastocysts, they didn't see any association with ploidy or aneuploidy. So the chance of being euploid or aneuploid is the same as if the embryo didn't have those cleavages, as I explained to you, because all these are with blastocysts. And regarding all the time cleavage parameters, 18 studies found out that aneuploidy was present, more present, when we had a delayed in these parameters that we see here in the left. And some others had no association, like time of pronucleus fading, that in fact we saw here in our clinic, but in this analysis they didn't see it. And also in these cell cycle durations. But the ones that I would like to tell you that were the most notable delayed variables in the aneuploid embryos were time to 8 cells, time to 9 cells, time to blastulation, and time to expanded blastocyst. So in case you are still building your model or something, you might focus on those and check them first, maybe. Again, there's a considerable variability between the euploid and the aneuploid embryos, making it impossible to classify them definitely. But we could use this idea to prioritize embryos to biopsy, for example. And since all the studies are very heterogeneous between themselves, again, we should think about another way to design these studies to be more robust and tested in-house. So this is a possible classification model that has been proposed, that you could just see which ones had which parameters that were delayed, and then you could prioritize them, but not necessarily one classification model is applied to all the groups, as we see in the other study on the right, that they didn't find the same result. And a little bit of curiosity about the morphokinetics and the ovarian age influence. So Dr. Thais told already that the maternal age influences the quality of the embryo, not only the ploidy. So if you have an euploid embryo from a younger patient or an older patient, the quality of that embryo is different. And we see that also in the morphokinetics. So if you have an oocyte from a younger patient or an embryo from younger or older, you might not see the differences in the same parameters. Specifically between the patients from 33 and 36 years, between euploid and aneuploid embryos, this group didn't find any parameters that had a significantly difference between them. And in the other groups, the parameters that they found a difference were different. In our clinic, we also looked into that, and we found out that in patients younger and older than 37 years, the time to blastulation was significant for both age groups between this ploidy analysis. And talking about the clinical pregnancy, the morphokinetic parameters correlated only in the patients younger than 37. Well, the mosaics, they are also embryos that have a different morphokinetic signature than the euploid and the aneuploid. Some groups have found that low-level mosaics and euploid embryos, they have comparable morphokinetic parameters. And the high level, they have some delays. Other groups found no difference between low and high level mosaics. And some other ones studied even a little bit deeper and found out that whole chromosome mosaics and segmental chromosomes, they also have some different parameters. And that should be taken in consideration when we try to evaluate that for ploidy. What about the sex chromosomes? Can we see if an embryo is a male or female just from the morphokinetics? Actually, the male embryos, they are a little bit faster in achieving the 8-cell stage than the female ones. We saw that here in our clinic, and it's also what other groups found out. And it's possibly because the female embryo, the female embryos, they have to inactivate the extra X chromosome. So that would be the explanation for this little delay that they have when compared to male embryos. And well, what I would like to tell you is that there is indeed some relation between ploidy and probably implantation and clinical pregnancy, since they are also all correlated, and morphokinetics. It is undeniable that the cell cycle, the genetics of the cells, they will reflect on how fast an embryo cleaves. And if we can see that on the opposite way, so from the cleavage time, understand what is the prediction of the ploidy implantation or clinical pregnancy of that embryo, is a little bit more complicated, but we should actually. And there are many variables, as I just said, tons of them, the maternal age, the chromosome of the sex chromosome of the embryo, these direct cleavage that might happen. So not to mention much more, the concentration O2 that is used in the incubator, the media, so where you live, the altitude, and there are many things that can influence on what we find, and we might need some help to put them all together. That's where comes the artificial intelligence. That's the whole idea, to put things together. I know that I'm a little bit beyond my time. I will try to rush here, but just to tell you a bit about the artificial intelligence, to build an algorithm, it demands a lot of data collection, processing, and then we have to train the model, validate, and applicate it. But the whole idea is to, as I just said, put together many ideas, many things that we wouldn't be able to do it by ourselves. And it could be applied in not only embryo selection, but also many other sectors of the artificial reproduction laboratories. And I would just talk a little bit more about the embryo selection and about how, from what we see, we could be seeing more or know more. When we apply some filters, when the artificial programs apply some filters, they don't extract only 3 variables of an embryo, like we would say from the one in the left, that we would only characterize how big it is, how the inner cell mass and trophoctoderm is. But actually, applying some filters, more than 275 variables could be extracted, like in this other image. And in the last years, many softwares have been published and applied. Actually, they are already available to buy and use in your clinics. And all of them go to embryo selection. So some of them is for ploidy, some of them is for implantation, some of them is already for clinical pregnancy, but they are always focused on the embryo selection. And the input might be very different. So you can try to predict ploidy status from morphology, or implantation from morphokinetics, or clinical pregnancy from the patient's information. Or you might also do a combination of them and put them together with the AI. We have started a study here in our clinical project that was the first one in Brazil in an AI ploidy model research. And this is the study that we have been presenting in the biggest conferences in the world. And we have been achieving results comparable to the ones published by other groups. So from morphokinetic parameters or from morphology, we are trying to predict euploid and aneuploid embryos, and I will just try to tell very quick how we have been doing it. So one example that I will use will be this one from a single image. So just from one picture of the embryo, can we predict if it is euploid or aneuploid? So from one image, the program extracts 33 variables because it applies multiple filters, and from the grayscale, the texture of the picture, it extracts some mathematic variables that are these 33 variables that will be used as the inputted ones in these artificial neural networks. So we give a picture, the picture is processed, there are 33 variables that are applied in the A and N, and then we give them the result to train it and then test later if he learned it, which ones were the euploid and the aneuploid ones. So there is a division of all the embryos that are provided. And then after all this training, validation, and testing steps, there is also a blind step. And that's where we have the final results to have an idea if it's working fine or not. We have been achieving something around 70% of correct results. That was the ones that I said that it's comparable to other groups' results. And this is how we see the result. So from the training, it learns really well. We see the rock curve that is almost perfect, so 0.98, 0.99. And then when we come to the blind test, the curve is of course not as good as the training one, and it shouldn't be that perfect, but we achieved something like 0.72 and 0.74. And once again, it's not perfect, but it could guide us to a model of risk classification. So again, once we have something, we have to validate it. And switching from ploidy to clinical pregnancy, we also have been doing this kind of study. So giving an image to the AI and asking it to predict the clinical pregnancy. And to know if the AI is doing good or not, we had to compare it to the embryologist, because that would be our gold standard, right? So does the AI knows more than us? That was the study that I presented this year in the ASRM. And just quickly, so that you could see, we gave the image to the embryologists and asked them if this embryo that was the best one that was chosen for transfer, if they thought that this embryo would result in a clinical pregnancy. And also gave that same image to the AI. And what we found out is that the AI was much better than the embryologist, especially in predicting the embryos that did not implant. So we can see in this graph that in the green line, every time that an embryologist was correct in the embryos that achieved a positive clinical pregnancy, well, actually they were very good, more than 55% in all cases. But for the embryos that did not result in clinical pregnancy, they were really bad, let's say it like that. And the AI was very consistent. So it was not so different when they said it was going to result in a clinical pregnancy or not, and it was much better in predicting the ones that did not achieve a clinical pregnancy. This is how we are doing nowadays. So we already have a software and we are ranking the embryos from a patient so that we can have an idea which ones would be the ones chosen for transfer first. And we are trying to do it prospectively now. And from the AI, my conclusion will be that the results that we have already give us confidence to incorporate the AI in the team of embryologists, trying to really improve the selection of the best embryo. And from the time-lapse system, my conclusion will be actually a question for you, because for me, there is benefit in using time-lapse system, and the most of it is the knowledge, and also all this whole new world that it brought to us, including the artificial intelligence applying the application of it in our routine and in the better selection of the embryos. I would like to thank you all. I'll thank you also for all the group here in Huntington and also UNESCO that are our partners in all this research that we have been doing. And also would like to invite you to our Congress that is going to be next year. So you are very close to us, please come. And I'm sorry I exceeded a bit of the time.
Muchas gracias, doctora.
Thank you, doctor, for your presentation. And thanks for the invitation, of course. A lot to learn, really. So we have a few questions. We'll try to go deeper in some topics. I am going to introduce our invited guests. Mrs. Florencia Nodar, a clinical embryologist, director of the embryology lab at Cegyr. Welcome, Florencia. And Dr. Andrea Quinteiro Retamar. She's a gynecologist specialized in reproductive medicine with the National Academy of Medicine. She authored many papers, an active member of Samer Almer. She is the coordinator of the surrogate program at Cegyr. So welcome, Andrea. And we open the floor to questions. If you have any questions or comments, Andrea, you can start. Well, great presentations. Let me begin by telling you that, and you wanted to know what you can say that is new, and it's actually a great deal. And thank you for the presentations and for clarifying the situation. I have a question. A patient that doesn't get pregnant after several transfers and patients that frequently miscarriage, sometimes you see these patients year after year and you don't have any answers. And Thais, in your clinical practice, do you foresee the use of PGT and time lapse and receptivity studies? Could you use these techniques to reduce the number of patients with failed implantation? And could you use these techniques with all patients in general?
It's the question of the million for the next couple of years, decades maybe. So I think, Andrea, that we have two different diseases, the recurrent implantation failure and the recurrent pregnancy loss. The literature doesn't combine the same knowledge for both of them. Maybe we can apply some things for both, but not all of the things. What I think that the young patients are different from the old patients. Usually when we deal with the old patients and we do the embryo selection through biopsy, we achieve better results. And even if we don't achieve better results, we simply change for egg donation, we achieve the pregnancy. So it's clear that the oocyte quality is an issue for them, okay? But here or in the young patients, if we don't achieve the results with good quality embryos, we must look for the endometrium, in my opinion. But we don't have a clear evidence to look always for the endometrium only over the literature. So we need, as we always said, more randomized controlled trials from different groups to achieve the same results as the EV achieved. So I think we have to learn more, maybe from this organoids. I like it very much in Nursery. This talks about organoids, the assembloids, the blastoids, and we need to understand better the crosstalk between the embryo and the endometrium. So I think this is the future, maybe.
Gracias. Excelente. Flor, ¿vos tenés alguna pregunta para Thais o no sé si le querés preguntar a
Any
Catherine?
questions, Florencia, for Thais or for Catherine? Yes, I would like to start with Catherine. Thank you so much for such great presentations and for your generosity with us and for sharing all the details with us, like Catherine did. So to answer your question, Catherine, of course, I believe the artificial intelligence is here to stay, is a great help for labs and for ourselves, because ultimately what we want is for the patient to take the baby home with her. So everything we can do to choose the best embryo to achieve our result in the shortest time possible, everything is helpful. This is our job, and IA is here to stay. And we just started this year using the embryoscope. We take down every data, we take a look at everything, but we don't know the use of all the data and what conclusions we can draw from that information. And we can spot early cleavage or late cleavages or reverse cleavages. So we are taking baby steps, we are learning. But given the results of all the groups, well, there's no agreement, there's no consensus on what is the best approach. So PGT is necessary to determine whether an embryo is euploid or not. So what is your experience with AI, with the euploid nature of the embryo and the correlation with PGT?
So regarding your question, the moment that we are in the artificial intelligence for the clinical pregnancy prediction is a little bit further than the plugin. So in this moment, we are still trying to find out what is the best combination of inputted data. So only the picture, or so only morphology, or should we also add morphokinetics that we actually already saw that it's I think it's the most important one. Or if we should also add some information about the patient and which information are the relevant ones. So BMI or the patient age, of course. So we are trying to see which one is the best combination to give us that prediction. And then we would have to come to the point of what you just said. Try first with the AI, see which result we are going to have, and then compare it to the real result. Yeah. So we are still in that step, but from what we have seen, we achieved a better pregnancy prediction with morphokinetics and the morphology as an input. So a single image of the embryo and the morphokinetic parameters. With that, we achieved 85% of correct results. So that's really nice. It's something like what we have seen with the non-invasive PGTA from the media. So if that's what's going to happen, that will be incredible. We could use it for the egg donation program that are patients that don't have this indication of doing an PGTA, and try to start with that line of thinking. But that was a really nice question. I'm happy to share a bit about it.
Vos, Thais, ¿qué nos podés decir de esto? ¿Qué pensás de seguir haciendo PGT?
What about you, Thais, about doing PGT to our patients until we can finally
Poder elegir el mejor embrión.
know, or we can finally choose or
PGT
select the best embryo? So, of course, what we want is to not touch the embryo so much, and that everything is much more natural.
Yeah.
So what's your opinion?
Thank you for sharing with me this embryologist side of the embryo view. But I think the same, and I'm with actually not with Catherine, but I'm receiving the embryologist results, and I think it will be the future, because that's why I put in the end of my presentation. I think less we manipulate the embryo, better for our patients' expectancy and results. So expectations and results. So I think I always like to talk about egg donation as a major endpoint for this analysis, right, Catherine? but of course, also the younger patients. As Andrea asked me, the young patients that we do not have higher chances of aneuploid embryos, we must select them without invade to be with less invasive methods. So I think it's a great thing for us if we achieve the consistent and right results that it can be replicable for all embryologists. I'm sure that will help us.
Yo le quiero hacer una...
Creo
Ay,
que el
perdón.
camino va por ahí.
I think that that's the way to go.
Sí, yo opino lo mismo. La verdad que
I
la gente
think
que
the
pasa
same.
por
Patients that go to donation after several failures,
es importante
I think it is important to offer them the
para seleccionar
best
un
tool possible to select embryos from a young patient
la tasa de
where you know that aneuploidy rates are very low, but also we need to try
posible.
to achieve pregnancy as soon as possible, especially in patients that come from several failures and they have been with the
No, muchas veces-
infertility for several years and they need an answer. And sometimes you get the feeling that we could do something more because whether you want it or not, the pregnancy rate goes up. With the blastocysts, it went up, and with PGT for elderly patients, it also
Siempre queda
went up.
porcentaje que
But there's always a percentage of women that cannot achieve the results, and you always want to get to 100%, and sometimes it is difficult to
con
do that.
respecto
But I'm sorry, I have one question for Dr. Catherine regarding the implementation of time lapse and how long would you say is the learning
cuáles son los
curve
mejores datos
to know what are the best data to consider
antes
for
del medio
analysis
en el
and
que
how we can help our patients in the meanwhile while we are doing the analysis?
Thank you for the question, Andrea. It's very relative, the time, because I think it's more about the amount of data that you have than actually the time. Of course, the time will be at least 1 year because you have to have the first born babies from that culture. So that will be the at least that time, I would say, but you have to have some data so that you can actually do that analysis that I told you. We started doing it with 100 embryos, or maybe even less, just to see what we could really see as a different parameter that was making some difference. So I couldn't say you the answer, the perfect answer, the right answer, but as soon as you start having it, you can, the live births, you can start having your sheet. And I would say that do it all prospectively. As Florencia said, that they are doing all the annotations, that's the ideal, because if you have to go back and see what happened with the embryos, that's terrible. Increases a lot the amount of work. But if you have that in mind already, so you can have your sheet and put it all down, so you can also track what is happening with which embryos. That is really nice. And you can start even from the moment that you have the embryo quality. That's what we had. So we just knew which embryos were A, B, or C, and we tried to compare it with the times that we had in that development of that embryo. So that's also already a start.
Buenísimo, gracias.
You're welcome.
Great, thank you.
Bueno, ¿hay alguna pregunta más?
Do we have more questions from
Nuestras especialistas.
our specialists?
Creo que no.
I think we have some
¿No?
Sí, pero
questions from the audience,
con las
but
preguntas
they
de
have
ustedes,
been chatting because these are based on your questions.
Similares.
So we don't want to repeat the questions because they're quite similar. They were following our conversation and chatting. So I think that we will say goodbye after such a very productive meeting. Yes, we are over with the agenda. We want more because we enjoy it, but the time is up.
Mi parte,
So
agradecer.
at least on my side, I would like to thank you all very much, Dr. Thijs, Dr. Catherine, for sharing your knowledge and your time with us. Also, Dr.
Que me acompaña,
Florencia, Andrea, and
como siempre, al
Monteiro,
público que
and
nos
Andrea
acompaña en esta
Cosia, who are with us today, and all of the participants who have joined our sessions. We learned a lot from this. To the professors at Cegyr, our technical support, and the industry, of course, who is sponsoring us so that we
este
can make
2022.
this possible. And throughout 2022, we've had 4 sessions. This is the last one, but you will have news
Que el Cegyr les va a
from
ofrecer.
us soon because we will have new webinar offers for you. So this is all on my side. Thank you very much.
Por ser
Thank
tan
you all so much for being so
Gracias.
generous with us.
Ha sido un
Muchas
placer
gracias.
escucharlos.
It
Un
has
placer
been
para
our
escucharlos.
pleasure. It has been our pleasure. Thank you.
La verdad, excelente.
Good afternoon. Brilliant, brilliant presentations. Thank you so much. Well, we will see each other soon then. Bye-bye.